welcome to Yaqeen Neet English Patch. Good afternoon, how are you doing? Let me know if the audio and video are both clear. There was like a slight 3-minute delay. I'm so sorry, I kept you waiting. Good afternoon everyone. Vidya, Pratik, Kirti, Arpita, Shravani, another Shravani, Rupreka, Satyam and everyone. Good afternoon. How's everyone doing? Do you have any doubts from yesterday's class? Yesterday we discussed the pigments, the different pigments, different kinds of pigments, what are its preferred wavelengths of light, all of that. Do you have any doubts? I want to increase the size of this. Just 1 minute. Okay, just give me 1 minute. I will fix this thing here. I'm not able to increase the font size. Just 1 minute. I'm getting it sorted. Notes is done, everything is done. Okay, fine. Full form of chlorophyll A. What full form does chlorophyll A have? Chlorophyll is not an abbreviation. Chlorophyll is the name of a pigment. सर क्या आप फॉन्ट साइज को इंक्रीज कर सकते हैं ये चैट का? और भी बड़ा. हाँ अब ठीक है. हाँ अब ठीक है. थैंक यू. या all right, everything is sorted. So what are we going to learn in today's class? We will learn in detail about the light reaction and how exactly during light reaction ATP synthesis takes place. Is everybody ready? Yesterday some people had doubts regarding the pigments used in the light reaction. I told you about the pigment complexes, what we call a photosystem, what is the reaction center, what is the light harvesting complex. So some people had doubts regarding light harvesting complex and they had pinged me on Telegram. Light harvesting complex is basically your antenna molecules according to NCERT. So in the photosystem, all the pigments other than your chlorophyll A reaction center, it's considered as the light harvesting complex or the antenna molecules or the accessory pigments. Is that clear? Cool. Let's move on then. I'll disable the comments for a little while. Pay attention, don't get distracted. All right. Cool. So what do we already know? From yesterday's class, we know that photosystems are of two types. There is PS1 and there is PS2. In the reaction center of PS1, you have chlorophyll A 700 that absorbs 700 nanometers wavelength of light. PS2 that has chlorophyll A 680. Right? We learnt about it. We also know about the location of PS1 and PS2. PS1 is present in your grana lamellae as well as your stroma lamellae. Whereas PS2 is present only in grana lamellae. So what exactly happens during light reaction? Let's understand that. During the light reaction, sunlight is utilized. Utilizing the energy of sunlight, it might seem repetitive, but I want you to remember what you've learnt earlier. It is captured by chlorophyll pigment primarily chlorophyll A, and it will produce energy rich molecules in the form of ATP and NADPH. Now in this light reaction, another thing that happens is water will undergo splitting or lysis and form oxygen, and it will also release some electrons and protons. More about this we'll study as we move on. So we know in the light reaction what is forming? An important molecule called ATP is forming. How is this ATP forming? ATP is adenosine A along with 3 phosphates. That's why we call it ATP. Now for the formation of ATP, adenosine diphosphate having 2 phosphates, ADP, adenosine diphosphate, will combine with an inorganic phosphate to form adenosine triphosphate. The process by which ATP is forming, this process is called phosphorylation. During photosynthesis, for formation of ATP, where is the energy coming from? The energy is coming from sunlight, right? So, this type of formation of ATP or this type of phosphorylation has a special name. We call it photophosphorylation. We have several other types of phosphorylation which we will learn in the next chapter. ATP forms in the mitochondria, ATP forms even in the chloroplast. Whenever ATP is forming in the chloroplast, as in the case of photosynthesis, by making use of the sun's energy, in that case, we call that process photophosphorylation. I repeat, phosphorylation is the process by which ATP is forming from ADP and inorganic phosphate. This process is phosphorylation. If this process happens during photosynthesis in the chloroplast by using the light's energy, we call it photophosphorylation. Alright? Now, let's learn about how exactly light reaction happens. What is the process by which ATP gets synthesized during light reaction? Okay. I told you that photophosphorylation is the process by which ATP forms during light reaction. Now this photophosphorylation happens in 2 ways. Can happen in 2 ways. Photophosphorylation can happen in 2 ways. The first type is called non-cyclic photophosphorylation. Pay attention. The second type is called cyclic photophosphorylation. In non-cyclic photophosphorylation, both the photosystems PS1 and PS2 are involved. In cyclic photophosphorylation, only PS1 is involved. Just give me 1 minute, I have to check something. All right. Cool. So these are the two types of photophosphorylation. First, we will learn about what happens in non-cyclic. Then we will learn about what happens in cyclic. Okay. So in non-cyclic photophosphorylation, like I already mentioned, both PS1 and PS2 are involved. Why we call this non-cyclic? I'll explain to you. Pay attention. This is important. Now, where is this non-cyclic photophosphorylation happening? It is happening in the thylakoid, right? In the thylakoid membranes, you will find the presence of PS1 and PS2 both. Okay? So this involves both PS1 and PS2. So over here, both of them are involved. But during non-cyclic photophosphorylation, it occurs in series. Functionally, PS2 one comes first, then PS1. Okay, both of them are involved. What is this? This is a thylakoid, a single thylakoid. That is where both PS2 and PS1 are present. Within the thylakoid, you have the lumen. Outside the thylakoid, you have the stroma. This right here, this membrane right here is the membrane of the thylakoid. Okay, so even for PS1 and PS2, on one side there is lumen, the other side, outside there is stroma. This is the location. But I will represent it to you schematically and show what exactly happens during non-cyclic photophosphorylation. First, pay attention, listen carefully. Okay, this is more like a block diagram. PS1, PS2, both of them are involved. So first, let us take both PS2 and PS1. I told you the sequence in which electron transport happens has PS2 first, then PS1. Then why did we call PS1 PS1? Why did we call it 1? Because that is the photosystem that was first discovered. Okay, that's the reason we call it PS1. So over here, I will have both photosystems. Let's say this is my PS2 and here I have PS1. Okay, so this is PS2 that has a reaction center of chlorophyll A 680. This is my PS1 that has a reaction center of chlorophyll A 700. Now, sunlight will be incident on both of these. Now, these are located in the thylakoid membranes. Where are these thylakoid membranes present? They are present inside the chloroplast. Where is the chloroplast present? Chloroplast is present in the mesophyll cells. Where is the mesophyll present? Mesophyll is present in the leaf. So when the sunlight hits the leaf, it hits the mesophyll cells, it hits the chloroplast, it hits the thylakoid membranes, and simultaneously on both PS2 and on PS1, light will be incident. Right? Most people think, most students get confused that first light will hit PS2, then it will hit PS1. That's not the case. PS1 cannot keep waiting. Just think about it. When sun is hitting the leaf, it is obvious that simultaneously on both PS2 and PS1 light will be incident, right? So that is what happens. So this on PS2 light will be incident, sunlight. On PS1 as well, sunlight will be incident. What happens in PS2 and PS1? In photosystem 2, photosystem 1, both photosystems, though chlorophyll 680 is the primary pigment, it is the reaction center, we know that surrounding it there are many other antenna pigment molecules. Depending upon which molecule it is, depending upon which wavelength of light it prefers to absorb, it will absorb that particular wavelength and pass on that energy to chlorophyll A molecule. Chlorophyll A will absorb 680 nanometers, chlorophyll A in PS2 will absorb 680 nanometers, chlorophyll A in 700 will absorb 700 nanometers. as a result of getting the energy from the sun's radiation. I showed you the structure of chlorophyll in yesterday's class. If you remember, I told you there is a magnesium present in the center. In chlorophyll A, there is magnesium in both chlorophylls A. One electron from that magnesium will get excited, okay, over here as well as here. So what happens here is you can say 2 electrons from each will get excited and get accepted by an electron acceptor. Okay, so there are 2 electrons that are lost from the magnesium of chlorophyll A680. There are 2 electrons that are lost from magnesium of chlorophyll A700. Now these electrons that have been excited are picked up by electron acceptors that are very close in the photosystem itself. Remember when I told you what all the photosystem includes, I said in the photosystem there are pigments, in the photosystem there are proteins, in the photosystem there are certain enzymes, and it also has certain electron carriers, right? So the first molecule to pick up that electron that has been excited is called the primary electron carrier, and this primary electron carrier is a part of the PS2 itself. So first the electron gets accepted by, which color should I use? Green looks good. The primary electron acceptor. Wait, let me write its name. It gets accepted by a molecule that is called Photolysis. The molecule that first accepts the electron from chlorophyll A680 is called Photolysis. Similarly, the molecule that accepts the electron that is excited from PS1's reaction center is a protein that contains iron and sulfur. We call it an iron sulfur protein. In short, you can represent it as FES. Alright, this lost electron that was captured by Photolysis. Photolysis took it up. This lost electrons after getting excited, and that was taken by FES. Now, both FES and Photolysis are called primary electron acceptors. Now, over here, whatever movement of electrons have happened, we say that this movement or transfer of electrons have happened uphill. This has happened uphill. Now what does that mean? That means chlorophyll A680 has a lower redox potential compared to Photolysis, which has comparatively higher redox potential. What is redox potential now? Redox potential is basically the tendency that a molecule has to accept or lose an electron. If electrons have to be transferred from a molecule that has lower redox potential to a molecule that has a higher redox potential, energy is required. Uphill movement will happen. Where is that energy coming from? That energy is coming from sunlight. The sun, if not for the sunlight, electron from magnesium can never be picked up by Photolysis. Because there is sunlight, because of the energy of sunlight, the electron from magnesium got excited, it exited the magnesium atom itself, and then it got picked up by Photolysis. For this, the energy came from sunlight. Okay, so this has low redox potential compared to this. This has a lower redox potential compared to this. Now let's see what happens first to this electron that has been captured by Photolysis. Now this Photolysis will transfer that electron to a series of electron carriers. Which are those electron carriers? Let's see. The first one obviously is pheophytin. Pheophytin will transfer that electron to a molecule called plastoquinone. Plastoquinone will further transfer that electron to cytochrome B6. Cytochrome B6 will further transfer that electron to another cytochrome, which is called cytochrome F. Then it will transfer the electron to another molecule called plastocyanin. In short, you can call this PQ. You can call this plastocyanin. You can call this PC. Okay. Now, where is plastoquinone present? Plastoquinone is present right here in the membrane. Plastoquinone will be present within this membrane here. If you can think of this as the membrane, it's not a great illustration, but yeah, if you can think of this as the membrane, over here, this is the phospholipid bilayer. So this plastoquinone is kind of like a mobile electron carrier that will be present here. Okay, so the plastoquinone will take it up. Now this plastoquinone that is there, which we are calling PQ, this is actually a hydrogen carrier. As it accepts the electron from here, it will simultaneously pick up protons from the stroma. As this plastoquinone transfers its electron to the next cytochrome, this proton will be pumped into the lumen. Okay. Now, I told you after plastoquinone, you have cytochrome B6, you have cytochrome F. Now, this cytochrome B6, cytochrome F, both of them are present as a complex. They are present together. Similar to how in PS1, PS2 you see multiple pigments, multiple proteins forming a complex structure and being present within the membrane. These two cytochromes, these are iron containing proteins. Okay, they act as electron carriers. They are of two types here in photosynthesis, cytochrome B6 and cytochrome F. Both of these will form a complex. They are present within the same complex. Alright. They are present within the same complex. And that complex is right here within this same membrane. This is the cytochrome B6F complex. Within the cytochrome B6F complex, you obviously have cytochrome B6 and then you have cytochrome F. So this plastoquinone will transfer the electron to cytochrome B6 then to cytochrome F. As it is transferring the electron here from, as the electron is getting transferred from plastoquinone to cytochrome B6F complex, it would have taken proton from here, right? It's actually a proton carrier. But our pheophytin is forcing this plastoquinone to take up the electron. It took the electron from pheophytin But since it's a hydrogen carrier, it will also pick up protons from the stroma side. Now, it will give its electron to the next electron carrier cytochrome B6, and whatever proton it had picked up from the stroma, it will throw it inside the lumen. So, when the transport is happening from plastoquinone to cytochrome B6F complex, protons are pumped from the stroma side to the lumen side. Okay? So that happens. Then cytochrome B6F complex has the electrons. It will further transfer the electron to another molecule called plastocyanin. This plastocyanin also is a mobile electron carrier. It will be present near the inner side of the membrane itself. It will kind of shuttle between these two. It will take this electron, pick it up, go to PS1 and pass on that electron to PS1. So what happens so far with respect to PS2? All of the pigments in PS2 captured whatever wavelengths of light that they prefer. The energy in the antenna molecules was passed on to the reaction center, which is chlorophyll A680. By making use of all of this energy, the magnesium in the chlorophyll molecule, one electron from the magnesium atom at the center of the chlorophyll molecule will get excited. This electron is picked up by the primary electron acceptor or the primary electron carrier, which is part of the PS2 itself, and that is called pheophytin. Now this has happened, this transfer of electrons is said to be uphill. It has happened from a molecule having a low redox potential to a molecule having a high redox potential. How is that even possible? For that, energy is required. Where did that energy come from? It came from the sun's radiation. Alright, now this pheophytin will pass on that same electron to a series of electron carriers. Over here, whatever movement is happening, this is happening downhill. Whatever is happening here, this is happening downhill. And when the electron is getting passed on from one electron carrier to another, the energy is getting reduced. It's becoming, the energy that it's carrying is becoming smaller and smaller. Okay, so plastoquinone to cytochrome B6F complex to plastocyanin, and ultimately the electron is passed on to PS1's reaction center, which is chlorophyll A700. As the electrons are being transferred through the cytochrome B6F complex, a molecule of ATP will get synthesized. Over here, ADP will combine with inorganic phosphate to form ATP. Remember when I told you that during light reaction, ATP will get synthesized? This ATP is getting synthesized as the electrons are getting transported through the cytochrome B6F complex. Alright, how exactly it forms, I'll explain to you later. For now, is this much clear? I don't even see any emoji option here, so there's no way of asking you. I have to finish this and only then ask. Alright, let's see what will happen with respect to, just give me 1 minute. So I was saying that the electron is picked up by this. Now listen, chlorophyll A700 also absorbed the sun's energy, and the electron from the magnesium in chlorophyll A700 also got excited. That was picked up by a primary electron acceptor, which is FeS, iron sulfur protein. Now this will again transfer it to a series of electron carriers. Which are those electron carriers? Next we have something called pyridoxine, which is represented by Fd. This pyridoxine will transfer it to NADP plus. Okay, I'll tell you what NADP plus is, what exactly will happen. And that NADP plus will accept the 2 electrons from pyridoxine. It will take up 2 protons from the stroma and form a molecule called NADPH plus H plus. NADP plus is oxidized form of NADP. It will accept 2 electrons from here. It already has a positive charge. By accepting 2 electrons, what will it become? By accepting 2 electrons and 2 protons, what will it become? It will become NADPH. It already has 1 positive charge. 1 electron is used to neutralize that positive charge. It becomes NADP with 1 electron. It takes another electron and another proton and forms NADPH. Both the electrons are used, 1 proton is used. The other proton will remain as H plus. Are you getting my point? NADP plus will accept 2 electrons from where? From pyridoxine. It will take up 2 protons from where? From the stroma. It will make use of both the protons and both of the electrons and 1 of the protons to form NADPH. And along with it, 1 more H plus will remain. That's why we write it as NADP plus plus H plus. Now, for the formation of NADPH or for the reduction of NADP plus into NADPH, an enzyme is required. That is called FNR. What does FNR stand for? FNR stands for pyridoxine NADP plus reductase. Okay, this is the name of the enzyme. It's not an electron carrier. PS2, PS1, simultaneously electron will get excited by absorbing sunlight with the help of antenna molecules. 2 electrons that left magnesium will be captured by the primary electron acceptor. Which is the primary electron acceptor for PS2? It is pheophytin. Which is the primary electron acceptor for PS1? It is FeS, which is an iron sulfur protein. Both of this is happening uphill. Electron is being transferred from a molecule that has a lower redox potential to a molecule that has a higher redox potential. And for that, energy is used from sunlight. Now, whatever electron transport will happen, that will happen downhill. Okay, over here also it's downhill, over here also it's downhill. So, from pheophytin, electron is transported to an electron carrier that is called plastoquinone. In short, we represent it as PQ. Now, this plastoquinone is a hydrogen carrier. It for sure accepts the electron from pheophytin, but simultaneously it will also pick up protons from the stroma along with the electrons from pheophytin. Once it passes on the electron to cytochrome B6F complex, whatever proton it had taken from the stroma, it will not release back into the stroma, it will push it into the lumen. Okay, so proton will get transferred from the stroma side to the lumen side when electron is being transported or transferred from plastoquinone to cytochrome B6F complex. From the cytochrome B6F complex, it is transferred to plastocyanin, which is represented as PC. The plastocyanin will transfer the electron to the reaction center of PS1. PS1 had lost its electrons to iron sulfur protein. That will pass it on to pyridoxine. That will pass it on to NADP plus, which will accept the 2 electrons from pyridoxine, pick up two protons from the stroma, form NADPH plus H plus. Over here an enzyme is required. You can simply call it NADP reductase, or you can call it ferredoxin NADP plus reductase. Alright. So if you notice here, chlorophyll A 680 lost 2 electrons. Chlorophyll A 700 also lost 2 electrons. The electrons that were lost from chlorophyll A 680 were used to replace the electrons that were lost from chlorophyll A 700. This lost 2, it got back 2. Photosynthesis must happen continuously. It's not like one cycle happens and it stops, right? For continuous synthesis of food, continuously ATP is required, continuously NADPH is required. So for this process to continuously happen, chlorophyll A must get excited multiple times. For it to keep getting excited, whatever electrons have been lost, that has to be replaced by one way or the other, right? So over here, there is no issue. It is sorted. It lost 2 electrons. It got its 2 electrons back. But over here, it lost 2 electrons, and the electrons from PS1 or PS2 did not replace the electrons lost from here. This is where the role of water comes in. I told you that water will split and form oxygen, electron and protons, right? That happens. Why does that happen here? In order to replace the electrons that PS2 has lost. If it keeps on losing electrons, will chlorophyll A molecule remain stable? Will it be able to do its job? It won't be able to do its job. For it to do its job effectively, efficiently, the electrons that have been lost must be replaced. In order for that to happen, something called photolysis of water takes place. Alright. For photolysis of water, during photolysis of water, what happens is water will split and form 2 electrons, 2 protons and 1 atom of oxygen. The 2 electrons that are formed here are used to replace the electrons that PS2 had lost. Once more this reaction happens, you get another oxygen atom that will become an oxygen molecule. It will be released outside the plant. These 2 protons actually have some role to play, about which we will talk later. Is this much clear? Why am I calling this non-cyclic photophosphorylation? I am calling this non-cyclic because whatever electron was lost from PS2, it never cycled back to PS2. Whatever electrons were lost from PS1, they never cycled back to PS1. So, it's not cyclic. The electron is not returning to the same photosystem that it was lost from. That's why it's called non-cyclic photophosphorylation. Why are we calling this photophosphorylation? In this process, ATP is produced. What gave the energy for formation of ATP? Sunlight gave it. So, we're calling it photophosphorylation. Why non-cyclic? Because there is no, the electrons are not returning to the same photosystem. Okay? Okay, Ashwita. Yes, Kirtana, it will become proton deficient. We want it to become proton deficient. That's how we will get ATP. You'll learn about it in a little while. Yes, Kirtana, it will, 2 electrons will get excited from each photosystem. NADP plus is not present in granulamellae, it will be present in the stroma. What else do we have? Where is plastoquinone in PS2? It's part of the PS2. It's part of that complex itself. Actually, it's not very complex. Maybe you're learning it properly for the first time. That's why there's a lot of words thrown at you. You're feeling that way. If you like read it 2 or 3 times, it should not become so confusing. For whoever is saying it's confusing, I will repeat it one more time. But some of you might have questions. Plastoquinone or plastocyanin or cytochrome B6F, none of these are mentioned in non-cyclic photophosphorylation in your textbook. When they're talking about non-cyclic photophosphorylation, they're not talking about it. But if you look at this diagram that is given for explanation of how ATP is formed, this comes later. But if you see over here, in this diagram, they've used PQ, they've used PC, they've labeled it, they've used cytochrome B6F. That is why I have explained it to you by making use of all this. If you look at your NCERT textbooks, if I have a textbook here, I could show it to you. Okay, I do have a textbook here. If you see your textbook here, if you look at these diagrams in your textbook, you have just electron transport system mentioned here. You have just electron transport system mentioned here. This is a very old textbook, probably from 2015, I guess, I'm not sure. So in this textbook, this diagram is pretty different. Over here, they've not mentioned plastoquinone, plastocyanin and all of that, PC, PQ, cytochrome B6F, FNR, all of that is not mentioned here. But in your new latest textbook from 24-25, all of that is mentioned. It's shown here, but it's not shown here. So I want you to know how exactly it works here. All right. Could you see what I'm trying to show? ADP will be present, Ankur. One shot for what, Maliha? Thank you, Anu. Correct, you're right, Janani. Lakshmi hasn't understood. I will explain it to you very quickly. Okay, Tissot has a good question. He's asking why ATP is not required in uphill. What does ATP do? ATP provides energy. Instead of ATP, here whatever energy is present in sunlight, that is being used. ATP is not used here. Instead of ATP, an external source of energy is used, which is sunlight. Everything is happening in thylakoid. See, for non-cyclic to take place, you need both PS2 and PS1. Both are required. And PS2 is present only in the thylakoid membrane. So obviously this has to happen only in the thylakoid membranes. It cannot happen anywhere else. I will repeat one more time. For those of you who are confused, please pay attention. We have two photosystems, PS2, PS1. PS2 has reaction center. In the reaction center, chlorophyll A680 molecule. PS1 has in its reaction center chlorophyll A700 molecule. Light is incident on both of these photosystems simultaneously. All of the antenna molecules will absorb whatever wavelengths of light they prefer, and whatever energy is obtained from that, they will transfer it to the reaction center. The reaction center chlorophyll A also will absorb its own wavelength of light that it prefers. By making use of all of this energy, from the magnesium in chlorophyll A of both these photosystems, 2 electrons will get excited. 2 electrons will leave magnesium atom and escape. These electrons are accepted by electron carriers. Whichever accepts the electron from the reaction center first is called the primary electron acceptor, or you can call it the primary electron carrier as well. Now the primary electron carrier or acceptor for PS2 is pheophytin. Where is this pheophytin present? Pheophytin is part of the PS2 itself. It's very close to where the pigments and everything is present, so it will quickly pick it up. For PS1, there is a iron sulfur protein which we represent as FES that will pick it up. Okay. In some books, it might be given as letter A, but it's actually an iron sulfur protein, so it will pick it up. All right. Now, what happens is, let's understand how the electrons are transported from PS2. Pheophytin will pass it on to a molecule, an electron acceptor as well as a hydrogen carrier, which is called plastoquinone. This plastoquinone is accepting two things. It is taking electrons From the pheophyton, it is taking protons from the stroma. The electrons that it has picked up from pheophyton, it will pass on to cytochrome B6F complex while simultaneously releasing the protons into the lumen of the thylakoid. From the cytochrome B6F complex, it goes to PC, which is not Priyanka Chopra plastocyanin. From plastocyanin, it is passed on to reaction center of PS1. Alright, as electrons are being passed from plastoquinone to cytochrome B6F complex, you can see that a molecule of ATP will get synthesized. Okay, how exactly it happens, I will explain later. Alright, for now just remember, here is where ATP will get synthesized. Now, whatever electron was lost from PS1 is first primarily accepted by this iron sulfur protein that is transferred to a molecule called ferredoxin. This ferredoxin will transfer the electron to a molecule called NAD+. Now, where is this NADP plus present? NADP plus is present in the stroma. So, from the stroma, NADP plus will accept 2 electrons from ferredoxin. It will pick up 2 protons from the stroma and get reduced, and it will form NADPH plus H plus. Alright. So, the electron that was lost from PS1, that has been replaced by the electron that was lost from PS2. The electron that was lost from PS2 has to be replaced. How does that happen? Water will split by a process called photolysis. Generally, water is a very stable compound. It does not undergo splitting usually, right? But over here, photolysis of water will take place. Water will split to form 2 protons, 2 electrons and a atom, nascent oxygen. These 2 electrons are used to replace the electrons that are lost from PS2. Alright. So, in this process, sunlight was used, pigments were used, ATP got synthesized, NADPH got synthesized, water got used, oxygen got released. Tawfiq, you're asking there is one ATP, one NADPH are produced in the light reaction. You can consider it that way. But there are so many different books giving so many different numbers that it just confuses you. Let's not worry about the numbers for now because the data related to these numbers, how many ATP, how many electrons, how many NADPH is formed is very different and research is still going on. So don't worry about the numbers for now. Alright? You can say, for now you can say, one ATP, one NADPH, you can say that. Sanya is asking, I said I will share the Google link. Okay, see, I tried. I'll tell you, I feel like a very bad student, but I'll tell you, I tried posting the link on the community section. I tried posting the link in the chat section of our videos. Like after chat, you can comment, right? So in that comment box, I tried to type. 2 times I tried to type here and there. I got blocked. Like I could not type anymore. I got blocked and I don't know, I will have to figure it out what to do about it. Apparently we're not used to, we're not supposed to type links and all of that. That's the reason I got blocked. What I instead thought of doing is starting my own Telegram channel that is public so that you can type my name and you will get my channel and you can add yourself or subscribe to that channel. Over there I will post the link. I already created it this morning. So if you have to look for me on Telegram, you will have to type and search this exact name, Shalini Somshekhar. You will see my picture holding a flower. So that is me. And over there I will paste the link of the Google form. Alright. See, even right now in this chat box, it is saying, I have been continuously trying to enter a prohibited word. You are blocked for 24 hours from chat, comment, doubt section. Please avoid, otherwise we will be blocked forever. Okay. Yeah, that happened. Alright. Let's move on. That's about your non-cyclic photophosphorylation. Now let's talk about cyclic photophosphorylation. In cyclic photophosphorylation, let's understand in what conditions it will happen first. Cyclic photophosphorylation requires, or it only PS1 is involved here. Only PS1 is involved. Okay. Where is a possible location where only PS1 is present? When we spoke about PS1 and PS2 in yesterday's class, and I reminded you of it today, I told you that PS1 is present both in the grana and the stroma lamellae. But PS2 is present only in the grana lamellae. So a location where you can find only PS1 is your stroma lamellae, because in grana lamellae you have PS2 as well as PS1. Where both are present in the grana lamellae, non-cyclic photophosphorylation will take place. I forgot to mention one point. This is also called the Z scheme. Okay, they should ideally be calling it the N scheme. I don't know why they want to call it the Z scheme, because it's kind of like zigzag like this, right? The way in which electrons are getting transported. Also, don't think this is like one route map Google Maps that electrons have and they're like, okay, let's go straight, let's go down, take 2 steps. This is not how it works. How this is represented, this schematic representation is based on the energy that the electrons are carrying. This has more energy, this has comparatively less energy, because every time the electron is getting transported, the same amount of energy won't be there. For the transfer itself, some amount of energy is required. So this is happening downhill. So based on the energy of the electron, this is like this. It's not like a route map for your electron. Electrons don't have brains. They can't think I have to take right, I have to take left. This is just chemistry, random molecules interacting with each other. This is definitely not a road map. Actually, how it happens is here. This is for your representation. This is for your understanding. We have a schematic representation. In the schematic representation, because it's zigzag like this, we're calling it the Z scheme. Over here, it is not like that. Understood? Now, yeah. Cyclic photophosphorylation, one thing is, it can occur in stroma lamellae, because over there only PS1 is present. What is the other option? I told you in the last class that PS2 will be functional only when it gets a radiation less than 680 nanometer. If it's greater than 680 nanometer, energy will be less. This chlorophyll A680 cannot lose an electron at that energy. Anything 680 or less, it still has the ability to absorb that energy. Over here, it is 700 nanometer or less, not more than that, because greater your wavelength, lesser will be the energy. Now imagine there is a chloroplast that has both PS2 and PS1 in the thylakoid membrane. If I'm incidenting this with just 700 nanometers wavelength of light, even though PS2 is there, PS2 cannot absorb it, PS2 cannot function. Even in that case, only PS1 will be functional. In that case, in the thylakoid membrane itself, PS1 can undergo cyclic photophosphorylation. Okay, for PS2, PS1 both to function together, you should have illuminated the plant with wavelengths 680 as well as 700. But if you're giving the plant only something that is beyond 680, then PS2 chlorophyll A680 will not be functional at that time. Even in the thylakoid membranes, though PS2 is present, PS1 only can function and it will undergo cyclic photophosphorylation. Alright, so here this can occur in the stroma lamellae, or it can occur in grana lamellae as well, but there is a condition. only when light greater than 680 nanometer is incident. Over here, only PS1 is present. Only PS1 is involved. So we have just PS1. This PS1 will have chlorophyll A 700. Sunlight will get incident on this guy. Okay. Now the electron from the magnesium of chlorophyll A 700 will be lost. That is picked up by what? That is picked up as usual by ion sulfur protein. Now what happens here is, instead of transferring the electron to ferredoxin and forming NADPH, it will take this route. What it does here is, this electron will get excited, electron acceptor will accept it, that will transfer it to cytochrome B6F complex and back to the same photosystem. This will transfer it to cytochrome B6F complex, cytochrome B6F complex to plastocyanin, plastocyanin back to PS1. Okay, so whatever electrons are lost from here is coming back to PS1. Because electrons were lost from PS2, the same electrons are returning to PS2, it's cycling back to PS2. We call this cyclic photophosphorylation. Similar to how when electrons were being transported through the cytochrome B6F complex, ATP was formed in non-cyclic. Even over here, when electrons are being transported through the cytochrome B6F complex, ATP will be formed. Since ATP is formed here, we call it phosphorylation, photophosphorylation. But the difference is it is cyclic. The same chlorophyll A 700 that had lost the electrons gets it back after it's going through all of these electron carriers. It's returned back to the same photosystem. That's why we call it cyclic photophosphorylation. Alright, so what happens here is, this will pass on the electron to, let's say, cytochrome B6F complex to plastocyanin back to chlorophyll A 700. Over here, one molecule of ATP will form. Because the electrons are being cycled back to PS1's reaction center, over here NADPH plus H plus will not form. Because the same electron is returning back to the same photosystem, there's no need for water to split. So over here, photolysis of water will not occur. That is the difference. Is it clear? Anybody has any doubts? Can you type the difference between cyclic and non-cyclic quickly in the chat? Once again, Neela Ganga. What is photolysis? Photolysis is splitting of water. Once again, Shravan also wants. Okay. Yeah, both can occur at the same time. What about PQ? Jatinder is asking, what about PQ? Good question. PQ is also there. Ferredoxin is also there. Okay. It goes from ferredoxin to PQ to cytochrome B6F complex. If you want, you can write it down. Do you want me to mention it here? Ferredoxin. Fine. Okay, I will repeat once more for those of you who are asking. Sunlight is incident. Electrons will get excited, picked up by FES, which is the primary electron acceptor. It will get passed to ferredoxin and then to plastoquinone, then to cytochrome B6F complex, plastocyanin, back to the same reaction center. Because plastoquinone, again, similar to how it is an electron acceptor and a hydrogen carrier, it will accept the electron, simultaneously it will pick up protons from the stroma and pump it into the lumen of the thylakoid. Okay, so ATP will get formed here. Now let's quickly differentiate between cyclic and non-cyclic photophosphorylation. This is non-cyclic and this is cyclic. In non-cyclic, both PS1 and PS2 are involved. Here only PS1 is involved. NADPH is formed. NADPH is not formed. Photolysis of water occurs. Photolysis of water does not occur. Oxygen is not produced. No, it is produced. Oxygen is not produced. Occurs in grana lamellae. Occurs in stroma lamellae. And when incident light is greater than 680 nanometer. Yes, Kirtana. Yes, Pratibha. Yeah, Sunetra has a point. Electrons don't return, electrons return. You can add that also. Electrons don't return to the same photosystem. That's the reason why we're calling it non-cyclic, right? So here electrons return to the same photosystem. Okay, I hope it is clear right now. We will move on if nobody has any doubts. Good job. Many of you have answered very well. Very nice. All right, we'll move on, children. Now I told you how during non-cyclic also ATP is formed. Notice how I'm drawing this when electron is being passed from plastoquinone to cytochrome B6F complex. Even in cyclic ATP is formed. Notice how I'm drawing it when electron is being passed from PQ to cytochrome B6F complex. All right. So we learn how exactly formation of ATP happens. That can be explained with the help of a hypothesis called chemiosmosis. Everybody knows osmosis, right? Or chemiosmotic hypothesis, you can say. This was proposed by a person called Peter Mitchell. So what basically happens in chemiosmotic hypothesis is, the hypothesis is that there is a proton gradient created across the thylakoid membrane. What does gradient means? What does gradient mean? Gradient means there is more of something on one side, there is less of the same thing on the other side. So if I'm talking about the proton gradient, I mean on one side of the membrane there is more proton, on the other side of the same membrane the concentration of proton is less. So there is a proton gradient. This proton gradient is generated during the light reaction. As a result of this proton gradient, ATP will get synthesized. Similar to how in osmosis there is a membrane involved, on one side of the membrane the water potential is more, or the concentration of water molecules, free water molecules is more. On the other side the concentration of free water molecules is less. Movement will happen from a region where it is more to where it is less through the semi-permeable membrane in osmosis, right? There we're talking about water molecules. Here we're talking about protons. So we call it chemiosmosis. Osmosis, something similar to osmosis where there is a gradient present across two sides of the membrane, but it's not of water, it's of some other chemical which is proton here. Okay. Now for ATP synthesis to take place, it is very essential that this gradient exists. Only if this gradient is there, ATP synthesis can take place. Because for ATP synthesis to take place, the enzyme that is involved in ATP synthesis, it derives its energy itself from proton movement. Okay, I'll explain to you how this works. For now, pay attention. This is very, very important. I want you to give like 200% attention, because in the next chapter, respiration, again, this is going to come. I'm not going to explain in detail over there. So the same thing will, something like 80% of it is similar. Even in respiration, over there I will not explain to you in detail. Alright, so what you're looking at here is just one single thylakoid. In the thylakoid membranes, you can find both PS2 and PS1. PS2 is represented by this block over here. PS1 is represented by this block over here. What you see as this yellow color motte or egg is the cytochrome B6F complex. All of these are present in the membrane of the thylakoid. You're already aware of it. I showed it to you in this diagram. There's PS2, PS1, cytochrome B6F complex. All of them are present in our thylakoid membrane, right? So they are there. Now, a proton gradient is generated, I told you. There will be more protons present in the lumen Okay, yeah. See over here you can see there is more protons present in the lumen compared to the protons in the stroma. In the stroma, the proton concentration is less. In the lumen, the proton concentration is high. What contributes to this difference in protons? What contributes to this gradient of protons that you see across two sides of the membrane? There are 3 points that we must understand here. Firstly, splitting of water. We'll start with that. Splitting of water or photolysis of water, this requires something called a water splitting complex. This will contain manganese and some other things. This water splitting complex itself will help water molecule to undergo lysis and release oxygen, protons, electrons. Now this water splitting complex is located over here towards the inner side of the thylakoid membrane. If this is located here, then obviously splitting of water will happen towards the lumen. If splitting of water is happening towards the lumen, protons, electrons, oxygen, all of that is produced inside the lumen. What happens to the oxygen? It diffuses, escapes and leaves. What happens to the electrons? It's passed on to PS2 to replace the electrons that PS2 has lost. What happens to the protons? It gets retained within the lumen itself. So these protons will stay in the lumen itself. Okay? Now, because of splitting of water, protons are getting generated. Protons are increasing in number in the lumen. One reason why there is more protons in the lumen. Let's understand the second reason. Just 1 minute. Okay, what is the second reason now? The second reason is, I told you that PS1 will transfer the electron from, the electron from PS1 will ultimately be transferred to NADP+. This NADP+ will for sure get electron from ferredoxin. But where will it get protons from? It will pick up protons from the stroma. So whatever protons are existing in the stroma, their number is further coming down because NADP+ is picking it up. Firstly, protons concentration is becoming more in the lumen because of water splitting. Secondly, proton concentration in the stroma is becoming less because NADP+ is picking up protons from the stroma to form NADPH. 2 points. This is the second reason. The third reason is, whatever protons are present in the stroma, that is being pumped into the lumen by the cytochrome B6F complex. Remember I told you, this plastoquinone that we have here, it is a hydrogen carrier. It acts as an electron carrier as well. From pheophytin, it will accept the electrons. From stroma, it will pick up the protons. It will donate the electrons, pass on the electrons to cytochrome B6F complex. But what does it do with the protons? It will pass it into the lumen, right? It will transport it inside the lumen. So whatever protons were present in the stroma, they're being pumped inside the lumen. Over here, the cytochrome B6F complex acts like a proton pump. Pump means active transport is happening. What does active transport mean? Active transport is when you're transporting something from a region where it's already less to a region where it's already more. Like you're pumping water against gravity. Waterfalls, for water to fall from the third floor to the ground floor, you don't require energy. But if you have to pump water from the ground floor to the fourth floor, you need a motor. energy is required because it's against gravity. Something that happens naturally, it's against it, right? So naturally, if you see, if you leave it as it is, molecules will move. Molecules will tend to move from a region where it is more concentrated to a region where it is less concentrated. But if you have to force molecules to move from a region where they're already less to a region where they're already more, that will require energy. All right. And whenever such a movement is happening, such a transport is happening, we call it active transport. Over here, there is active transport of protons happening from the stroma side to the lumen side. Because in the stroma, it's already low. In the lumen, it's already high. But despite that, we're moving it against its concentration gradient. That is done by the cytochrome B6F complex. That is why we're calling it a proton pump. Pump is only used when it's active transport. Otherwise, we don't call it a pump. Now, where is this getting its energy from? It's getting its energy to pump protons from this electron transfer itself. As the electron transfer is happening downhill, whatever energy it gets from this movement of electrons, it will use that energy and pick up protons from the stroma and pump it inside the lumen. Because of these three reasons, the concentration of protons in the lumen is greater than the concentration of protons in the stroma. I repeat, first thing, splitting of water is happening inside the lumen, so protons are released inside the lumen. Formation of NADPH is happening towards the stroma, so protons from the stroma are being used up. That is reducing proton concentration in the stroma. This is increasing proton concentration in the lumen. What is the cytochrome B6F complex doing? It is acting like a proton pump. It is pumping protons from the stroma side into the lumen side against gravity, actively transporting protons. As a result of all this, you can see that the concentration of protons in the lumen is more compared to the concentration of protons in the stroma. So what happens then? Protons will have to, if equilibrium has to be attained, there is some way they have to find out and come outside, right? You cannot forever keep on pumping protons into the lumen. For you to pump protons into the lumen during the next cycle, you need protons on the outside, right? If you keep on pumping it inside, all the protons on the outside will become exhausted. You will have no more protons. For this to happen continuously, protons must leave here, again get pumped back, pumped back, right? So what happens now? These protons have to move passively along the gradient from the lumen to the stroma side. It cannot move through this through the phospholipid bilayer of this membrane because they are lipids, right? Through this phospholipid layer, charged particles cannot move. Proton has a positive charge, it can't just diffuse out through the membrane. There is where this ATP synthase comes in the picture. ATP synthase also is a part of the thylakoid membrane. It is a complex. It has two components. One component is called CF0. The other component is called CF1. This CF0 component is right here. Okay. This is the CF0 component. This CF0 component kind of forms a channel in this membrane. Through that channel, proteins will come outside. Okay. As protons will come outside. The protons will move outside. How is it moving? It's moving along the concentration gradient right now. It does not require energy because it's moving from a place where its concentration is high to a place where its concentration is low. So it's moving along this gradient. Now, as the protons are moving through the ATP synthase molecule, you can say that a proton motive force is generated. Some force is generated. As a result of that force, in this CF1 component that you see here, this is called CF1. The same ATP synthase is present in your mitochondria also. There we call it F0, F1. Here we call it CF0, CF1 because it's part of the chloroplast. Alright. Now this CF1 is like a head. It has a head-like structure. You can think of it like a ball pin. Like it has a stick kind of part and then it has a head kind of part. This stick kind of part forms a transmembrane channel through which protons can pass. Now this head, you can think of its structure like a peeled orange. If you peel an orange, you'll have many subunits, right? So the head of this also appears similar to a peeled orange. Now this head will have many different conformations. Okay. As the protons are moving through this transmembrane channel that F naught has created, as the protons are moving like this, that energy will cause CF1 to change in conformation. Before that protons move through it, this CF1 is loosely holding one ADP on one side, one inorganic phosphate on the other side. As the protons move through it, it will combine these two. It will combine ADP and inorganic phosphate and release ATP. Where is it releasing ATP? It's releasing ATP into the stroma. Look at how this is oriented. The head is not present towards the lumen. The head is present towards the stroma. As the protons are moving through this, that will generate sufficient energy for this ATP synthase head to combine ADP and inorganic phosphate and synthesize ATP. Alright, because this enzyme, this complex is involved in the synthesis of ATP, we call it ATP synthase. Okay, so how is ATP getting synthesized? Because of the proton movement. What caused the proton movement? There was a concentration gradient, proton gradient across the membrane. What are the contributing factors for proton gradient? Splitting of water, pumping of protons, and using protons for the formation of NADPH from the stroma. These are the contributing factors for the generation of the gradient. But of all these three, the major contributor is the proton pump cytochrome B6F complex. Because of this cytochrome B6F complex pumping action, ATP synthesis has been made possible. That is the reason why, though there are many contributing factors, we are mostly considering this because this is the major contributing factor. Because of that, we always represent ATP synthesis here, and that indicates for ATP synthesis protons were required. Where did we get that protons from? This guy helped pump those protons. That's why we will represent it here. Apart from this, it's not like cytochrome B6F complex will take one ADP, one inorganic phosphate and make ATP. This guy is not directly involved in the formation of ATP. For ATP to be synthesized, ATP synthase alone can do the job. But how is this guy contributing to it? If ATP synthase has to do its job, a proton gradient must exist. This guy is helping in generating that proton gradient by pumping protons from one side to the other. Alright, that's the reason why you will represent it there. Doesn't mean this is forming ATP. Alright, now for the synthesis of ATP, there are certain prerequisites that you must have. There should be a proton gradient. There should be a proton pump. There should be a membrane. And there should be the enzyme ATP synthase. All of which are present here. There is a proton pump, a proton gradient is existing, membrane is there, ATP synthase is there. So ATP synthesis is happening. If anybody has any doubts now, you can ask. I think this is the first time I felt like I just started class and it's already over. Time really flew today. Kirtana, FNR can be present associated with the thylakoid. FNR enzyme is present in the stroma as well. Remember, NADPH formation is required for photosynthesis, but for some other reaction, NADPH may be required in the cytoplasm, in the stroma. So FNR is present in other places as well. It's also present in the thylakoid membrane. It may be present in the stroma as well. Anything else? Anybody has any other doubts? Please read it today itself. so that you will understand it very clearly. And tomorrow do we have a class? We don't. Next week, I will solve your doubts. Okay, for those of you who want the link, you know what to do. You can find me on Telegram. This is my name. Search the full name. You will get one channel. Subscribe. I will post the link there. There's no time for question today, Meghna. I don't want to simply rush to solving question. I want you to fully understand the concept and come for solving questions. Otherwise, I will be very disappointed. Take some time, understand the concept, come very well prepared for the quiz, and then you will solve all the questions. All right? So I think it's time and we can end today's class. I'll see you on Monday. Take care, everyone. Bye-bye. If there's any Zoom session that I will schedule, you will get the notification on the app. Okay? Bye.